Okay. All right. So we left off last time basically demonstrating the principle of segregation. We just mentioned on the quiz what the principle of segregation explains, but here we'll sum it up. Basically, the idea of the principle of segregation or the law of segregation. Adults have two alleles for each gene. If the two genes are different, we'd call these individuals heterozygous, one being dominant to the other. True breeding individuals have two copies of the same allele. They are homozygous, and this is what makes them true breeding. Every gamete they make carries the same trait, carries the same allele. And then the alleles segregate as gametes are formed. So just because an individual has the dominant phenotype does not mean that they can't make any gametes that bear the recessive allele, because, again, these segregate from each other. This was the dominant explanation against blending inheritance being a possibility. The fact that you're not just blending characteristics that you have two discrete factors that separate. Half of the gametes will have one allele and half will have the other, and that's the principle of segregation. Any questions here? We need to talk about test crosses today. We're not going to talk a great deal. We have a question dealing with dihybrid crosses. The mechanics of a dihybrid cross are very much like a monohybrid cross. In fact, if you want to make it really simple so you don't have to remember really any difference, you can do a dihybrid cross as two monohybrid crosses and then combine the two. All right? Anyways.
Are you recording this right now?
I am. Are you leaving right now? Okay, so our next question that we need to deal with is this: how does probability factor into genetics? And as we address this question, we're going to get an understanding of how we can figure out the probabilities of offspring carrying particular traits, regardless of what the particular traits are. All right. So this is a nice little illustration. It's a fairly simple one, although it's one that is extremely similar to anything inherited under a normal Mendelian inheritance pattern, right? You've got a coin, and if you flip a coin, it can land on one of two surfaces. It can either land heads up or it can land tails up. And just like if you have two different alleles for any one gene, your gametes can get one of two forms. Either get allele 1 or get allele 2. And so what we've done is we've set this up to where here's the probability for one set or for one flip, and it can be heads or tails, and each one should occupy half of the flips. And if you're going to flip a second coin, here's the setup for the second coin. Again, you have two options, each occurring half of the time. And then so now if you are going to flip two coins, we're going to combine these. And so the probability of flipping a head both times would be the probability of flipping a head once times the probability of flipping a head a second time. Okay, which would give you a probability of 1/4. Same thing for head and tail. Same thing for tail, then head. And then finally the same thing for tail, then tail. And so it's actually a really good illustration of what's going on with inheritance and formation of gametes, but tends to be a little bit, it's virtually the same thing, but it seems to be a little bit simpler because it's flipping a coin rather than constructing gametes. All right? We okay with this? All right. So looking at it in terms of the inheritance, if you have an individual, this one is actually right now. So here we have what type of cross? Looking up here, what do we have? Monohybrid cross. And how do you know it's a monohybrid cross? Both individuals are heterozygous. They have one big P and one little p, right? Big P, little p. This one is actually correct. The other diagrams, they had both little p's. But big P, little p, and you're only dealing with one character. So you have two heterozygous individuals and you're dealing with one character. So this is a monohybrid cross. And just like if you are flipping a coin, there are two options, and each of them are going to occur half of the time. So half of the time, this parent would produce gametes with the dominant allele, and half of the time it would produce gametes with the recessive allele. Same thing for this parent. And so if you want to know what's the probability of getting 2 dominant, it'd be 1/2 times 1/2, in the same way as the probability of flipping a head 2 times. Or what's the probability of getting 2 recessive alleles? It'd be the probability of getting a recessive allele the first time, which is 1/2, times the probability of getting a second recessive allele from the other parent, which is also 1/2, which gives you 1/4. And then the probability of this parent, usually the father is represented on the left, giving a recessive P is 1/2. The probability of mother giving a dominant P is 1/2. So the probability of this combination is also 1/4, 1/2 times 1/2. And then finally, the other, where the recessive came from the mother and the dominant came from the father, 1/2 times 1/2, and again, 1/4. But you can see that phenotypically, we're dealing with a different story, right? And even genotypically, because these two produce the same genotype. Now, the recessive allele may have come from the mother in one and the father in the other, but they both are heterozygous. So this is the same genotype, and this right here is a unique genotype, but heterozygous and homozygous dominant produce the same phenotype. So your ratios are a little bit different, but in terms of, so your ratios for phenotype are a little bit different than your ratios for genotype. Any questions there? Yeah.
Monohybrid because they're crossing, it's just one trait?
Yes, one character and they're both heterozygous. So a dihybrid cross would be 2 characters where they're heterozygous for both characters.
So the assumption is that they are the same in all other traits for monohybrid cross?
Yes, and if you're going to do a good monohybrid cross, they would be the same for all other characters. The only thing that would be different, so they would be true breeding for all characters except the one in question. The one in question, they are heterozygous. The other loci, they're all true breeding.
So they'd be homozygous for the other ones?
Yes, and they could be homozygous dominant or homozygous recessive. You don't care because you're focusing on the character in which they are not true breeding. So they may always produce green seeds, but they can produce both purple and white flowers. And they may always produce smooth seeds, but they produce both purple and white flowers. All right, so how does probability factor into genetics? We have what's called the product rule. And so if you want to figure out the probability of any particular genotype, you multiply the probabilities of the 2 gametes that fused to form that offspring. And again, this happens when 2 or more events are independent, like flipping a coin. Right? Just because you flipped a head doesn't mean it's any less likely that you're going to flip a head a second time. And just because you made a certain type of gamete does not impact the probability of your mate making a particular type of gamete. All right, then you can factor in the sum rule, and that's what happened when we were determining that there are 2 different ways to make a heterozygous offspring, right? The recessive allele could have come from the mother, or it could come from the father. So if you want to figure out the probability of getting a heterozygous offspring, you add them together because there are 2 different ways to get the same genotype. Right? Just like in flipping a coin, if you want to know what's the probability of getting 1 head and 1 tail, there are 2 different ways you could do that. You could flip heads first and tail second, or you could flip tails first and head second. Ultimately, it gives you the same thing, 1 head, 1 tail. So you'd add those 2 probabilities together. What's the probability of flipping head first, tail second? 1/4, right? Because the probability of flipping head first is 1/2. The probability of flipping tail second is 1/2. So the probability of doing that is 1/4, 1/2 times 1/2. What's the probability of flipping tails first and head second? 1/4. So what's the probability of flipping 1 head and 1 tail? 1/2, because now you would do the 1/4 plus 1/4, because there's 2 different ways to get the same result. And so what's the probability of 2 heterozygous individuals producing an offspring that is also heterozygous? 1/2. Okay? Same as flipping a coin. All right? Now, There are, yeah, you just have to remember that there are two ways to get the purple phenotype in a one character cross, and so you have to keep in mind the sum rule. Now, a Punnett Square is not necessary. There are ways in which you can do this without putting together a Punnett Square, but the Punnett Square is really helpful just for visualizing everything that's going on. So I will not give you a question on the final exam that requires you to draw a Punnett Square. However, I will give you a question on the final exam that asks you to determine the probability of producing a certain type of offspring. Okay, if you could do it without doing a Punnett Square, that's fine. But the Punnett Square is helpful for visualizing, because another way you could do it is just do the product rule, right? You figure out, okay, what's the probability of producing that type of gamete, and multiply it by the probability of the other parent producing the gamete necessary to generate that particular genotype. All right? Any questions, thoughts, concerns, things you feel like you want to share before we do a lecture break? Happy Friday, by the way. I know it may not feel incredibly happy, but it could always be worse, right? Yeah.
The Punnett Square, no, that seems rather simplistic. How many things actually follow? I mean, are there really usually just two factors for each zygote for a total of...
Oh, so are you saying are there usually only two alleles for any particular character?
Well, I guess there are. On one
With some characters, like Widow's Peak, you either have a Widow's Peak or you don't, right? So there's two characters.
How many characters in a single organism? I guess it would vary, but how many characters rely on that?
On a simple Mendelian pattern?
Yes.
It depends on the organism.
Okay.
In humans, it's relatively few characters are determined by simple Mendelian inheritance. Because relatively few of our characters are determined by a single gene that only has two alleles.
So how does the rest of it work?
More complicated interactions. Epistatic effects, polygenic effects, all sorts of wonderful things. We'll talk about that next week when we talk about human genetics. And we'll step it up a little bit. All right? What I want you to do for lecture break, I want you to get into relatively the same groups as those that you did the quiz, except for some people have disappeared following the quiz. So your group may be a little bit different. But I want you to go through it, and I want you to, without consulting the slides, and so you may have to rely on one or two people in your group, I want you to demonstrate how you can use a test cross to determine the genotype of an individual. Okay, how you can use a test cross to determine the genotype. Take a couple of minutes, work at this, and then we'll do it together. All right, take a couple of minutes, starting now. Yeah.
Would it be like multiple test crosses? Or one test
No, you can do it with a single test cross.
I'll pick the rules.
Oh yeah.
And when you go test cross, you get to see the alleles.
Yes.
You see if it's like B times C times B.
Yes. Yes.
Test crosses.
So we don't need to write with it.
And then all their offspring don't count if they're all different. We know that both of those individuals were, because basically just true breeding. Yeah. If you just get one that exhibits one phenotype and another, and you cross them until you can see if they're true breeding or not.
Are we
Oh, so you're trying to see if they're heterologous or homologous? Is that
Yes.
what you're trying to determine? Okay. Yeah. Then,
we're using a test cross to find
well, the test cross that were like the one that he was showing, the idealized one where you have a dominant recessive and a dominant recessive, and so you see, three of them display the dominant traits, even though two of them are not breeding.
Yeah.
Recessive, dominant, dominant. So, are you trying to explain like the differences of true dominant and maybe heterozygous?
Yeah, so it's to see whether or not those two individuals, like you're trying to figure out what those individuals are.
Got about 30
They
seconds.
exhibit the purple phenotype, you don't know if they're homozygous or not.
Well, in your test cross, you can see 50% of them. So if you choose them, it would be dominant. Right? Because that recessive trait carries out two lines.
No, if you think about
Right.
And then dominant. Right?
Alrighty.
And the other is dominant recessive.
Alrighty. What's that? Do we have some groups that want to share their thoughts on how you use a test cross to determine genotype?
Okay. So with a test cross, we're trying to determine the genotype of the parents.
Yes, and so you know, not of both parents, just of one parent.
Do you know the phenotypes of the parents?
Well, of course, you can look at them.
Okay. And then, so we can see the phenotypes of both parents, and we're trying to figure out the alleles. Or the genotype.
Yes. Yep.
Okay, so if you had one parent,
Uh-huh.
if you had both parents were expressing the dominant phenotype.
Yes.
And then you looked at the easy example I'm gonna coach, and then you looked at the offspring.
Uh-huh.
And it was demonstrating the recessive
Mm-hmm.
allele, then you know that both parents were heterozygous. 'Cause would
Yep, right, they have to be.
recessive.
Yep. And just one being heterozygous isn't enough to generate an offspring with a recessive trait. Right?
Because that D could.
Yep. Because and I'll show you that in a second. Usually what you do when you do a test cross though is you would cross a individual with the dominant phenotype. Like this example here, an individual with a purple flower, and you would cross it with an individual with the white flower. And the nice thing about that is, any individual with the white flower has to be homozygous recessive, right? There's no other option.
You're crossing a heterozygous with a homozygous when you're doing a test.
No, not necessarily. You don't know what the genotype of this individual is. You're crossing a dominant phenotype with a recessive phenotype.
So you know one of them's homozygous.
Yeah, you know this has to be homozygous recessive because that's the only way to get the recessive phenotype. This one you don't know. But then after your cross, you can figure this out very easily as long as certain things are true. And what has to be true in order for this test cross to work? You need to generate more than one offspring in every cross, right? Otherwise, it's like you have no idea what's going on. Although, unless you got lucky and you only generated one offspring and it happened to have the recessive character, then you would know that this individual had to be heterozygous in order to produce that. Okay, and so here it's demonstrating a cross between, in this case, this individual is heterozygous. We didn't know that going in. But if it's heterozygous, half of the offspring produced should have the dominant character because they picked up the dominant allele from that monohybrid parent's gamete. And then half of the offspring should have the recessive phenotype because they picked up the recessive allele from the monohybrid parent's gamete. And then here's what it would look like if that parent were actually homozygous dominant. 100% of the offspring would have the dominant phenotype because this parent can only produce gametes that have the dominant character. Okay, that have the dominant trait. Sorry, not dominant character. Dominant trait. And so every combination is going to generate you heterozygous offspring, which are all going to have the dominant phenotype. So you can only carry out a test cross if you know the genotype of at least one of the parents. I guess you could do it if you didn't, like in your example, Joey. You could do it if you didn't know the genotype of at least one parent, but you'd have to get lucky. You'd have to get lucky, and they'd both have to be heterozygous. Because if one was homozygous dominant and the other were heterozygous, all the offspring are going to be dominant phenotype. If both were homozygous dominant, all of the offspring are going to have the dominant phenotype, right? So, yeah, and then you also should do it with organisms where you get more than one offspring produced from every single cross.
What if both were heterozygous but dominant?
Then,
Would you still be able to see the
yep,
recessive?
yeah, but, so you'd have to get lucky if you didn't know the genotype of either parent. Right, but it happens, though. So we'll talk about human blood type next week. But human blood type, the only way to be type O blood is if you have two recessive alleles. And so if your parents both have a type A blood and you have type O blood, your parents have to be heterozygous. That's the only way in which you can get type O blood. Or maybe your parents aren't actually your parents, right? You could be adopted. Yeah, Anna. Okay.
But when you like breed those two flowers together or do two flowers together, sometimes they like will
Yes. So what happens there is you have something that's a non-Mendelian inheritance. You have a different type of inheritance, which again we'll talk about more next week. We'll talk about different modifications to Mendelian inheritance. If you get like a mixing of the two phenotypes, it's called incomplete dominance. Yeah. Ooh, thank you for that. So yeah, again, test cross, experimental cross, typically between an individual with the recessive phenotype, that's homozygous recessive, and any individual with the dominant phenotype. So it's used to determine if the dominant phenotype is homozygous dominant or if that individual is heterozygous. And so summing this up, if half the offspring have the dominant phenotype, the parent was heterozygous. If all of the offspring have the dominant phenotype or the dominant trait, then the parent was homozygous. Or your sample size just isn't big enough. Right? What's the probability of flipping a head 6 times in a row? To the sixth power, which is 1 over 64. Okay? Which is about just under 2% of the time. So if you flipped a coin 6 times in 100 different sets, about 2 out of every 100 of those sets should have 6 heads in a row. So yeah, the probability is fairly low, but it happens, right? So this is why you need lots and lots of offspring if you're going to say anything confidently genetically. Or alternatively, you could just sequence the genome of the individual and then you know what they are. Although that right now, I think the cost to sequence one of your genomes, one of your 2 genomes, is about $1,500. No, it really isn't. I mean, considering it cost them $3 billion to sequence one genome in 2001. So you could get both your genomes sequenced for about $3,000. The forecasts are that in the next 10 years, the cost of sequencing both of your genomes will be under $100. At which point, right. Well, I mean, it's the price of a nice dinner, right? For the price of a nice dinner, you can know the sequence of both your genomes.
What would be the benefit of knowing that?
Well, then you would know what's the probability of you producing offspring with any particular genetic condition. You could also know what propensity do you have to develop developmental or genetic conditions based on your genome. You could, right? And so you could do it backwards. And yeah, so all 3 of my siblings, well, one, we all look pretty much alike, and we all look, unfortunately, like our parents. And so, but we all have type A blood. And so we all have type A blood, so there's a pretty good probability that my parents have type A blood. And that they probably are not, at least one of them is not heterozygous. But there are only 3 of us, so they could both be heterozygous, and then that ratio, expected ratio of 2 of a monohybrid cross is 3 dominant phenotypes for every 1 recessive. Well, we don't have 4, right? So maybe the 1 recessive is waiting, you know? My parents are really old now, but not as old as Abraham and Sarah. So, yeah. Yeah, so like 23andMe and Ancestry.com, they are looking for particular markers. They're not sequencing your entire genome. They are sequencing millions of bases. I forget the exact number that 23andMe and Ancestry use, but it's in the millions. And they're looking at what are called single nucleotide polymorphisms, so places in the human genome where one base differs. So it may be a gene, it may not be a gene, but it's a known region in the human genome where I may have an adenine and you have a thymine, right? Or I may have a guanine and you may have a cytosine. So there's variation at that point. A lot of points in the human genome, there's no variation. Every single human being on Earth has the same base in that position, and a lot of the genome is that way because any modification there is catastrophic. It doesn't survive. And so what they're doing is they're looking at these suites of places where there are all those polymorphisms, and then they're using that to compare you to known alleles in particular regions. So if they're doing it based on, if you're looking at your ancestry, they're doing it based on, okay, well, 90% of the people that descend from the British Isles have this copy. And so if you don't have that copy, there's a low probability you descend from people from the British Isles. At least from, you have a significant number of ancestors in your lineage from the British Isles. And so on and so forth. And then they could do it for genetic conditions as well. They could say, 80% of the people that develop early onset Alzheimer's have this particular allele, and you don't. So you're not significantly predisposed to early onset Alzheimer's based on this marker. Yeah.
What's the probability of a child being male or female? Is that 1/2?
1/2, maybe.
There's times where like families have all girls or all
Yeah, it's not quite that simple because there are a number of ways. Our bodies tend to be a little bit more flexible with the sex chromosomes. So, for instance, you can have a male that has 2 Y chromosomes, and they could be relatively normal. Now, they're going to have a lower than average fertility rate. A lot of their sperm are going to be infertile because the chromosomal numbers are wrong. But they have a greater probability of producing male offspring because they have 2 Y chromosomes instead of 1. So 2 out of every 3 of their gametes are going to carry the Y chromosome rather than 1 out of every 2. So the probability is a little bit higher.
Are you talking about a YYX chromosome?
Or yeah, XXYY. Yeah.
Or versus a XXX?
Yeah, so triple X syndrome, triple X. And so all of these, with the sex chromosomes, abnormal chromosomal numbers, there are pathologies associated with it. And they tend to have lower levels of fertility than somebody with the normal number of sex chromosomes. But other than that, they usually are relatively normal and become fully normal with minor hormone therapy. So, yeah, and so you can have somebody that's XXY and they're still male because they have a Y chromosome, even though they also have 2 X chromosomes. And so you don't become male because you have only 1 X chromosome. You become male because you have 1 Y chromosome. Just in the same case, you can have a female that only has 1 X chromosome, doesn't have a second, and she will develop into a female. And it's not because she has 2 X chromosomes, she only has 1. It's because she does not have a Y chromosome. And really, she doesn't have just a small portion of the Y chromosome that leads to a shift in development. That's it? Okay. All right. So what is independent assortment? What is independent assortment? This was Mendel's second law, right? Law of segregation or principle of segregation, and the law of independent assortment or the principle of independent assortment. And you can explain this just based on alleles, or you can explain it based on chromosomes. Either way, you're explaining the same thing. And we already did this last time when I showed you the chromosomes of a monohybrid individual, and we did it with our brown hair, blonde hair, brown eyes, blue eyes. And independent assortment just means that all combinations are possible. Okay? And so if you look at independent assortment here, and so here, this is a cross to generate dihybrid individuals. And so you can see that this individual is a dihybrid because you have 2 different characters, 2 different genes. You have seed color and seed texture, really exciting characters there. But you have 2 different characters, and the individual is heterozygous for both. So this is a dihybrid individual. And if you want to know if these characters assort independently of each other, and what that means is that they don't come as a unit. So you notice one of the parents, and it doesn't label male or female, but we'll assume this is female because she comes first. And so this individual here is homozygous dominant for both, right? And this individual here, the male, is homozygous recessive for both. Really creepy looking seeds, right? And so if you use these individuals to generate a dihybrid, if these 2 characters do not assort independently, the only phenotypes you can produce are those that you started with. Okay? If these did not assort independently, you'll produce offspring that are either yellow and round or green and wrinkled. You'll produce individuals that either have the dominant phenotype for both or the recessive phenotype for both, but no combination of the two if they don't assort independently, because it means they're coming as a unit. It means that the chromosome that these individuals have, one from the mother, and what does the maternal chromosome look like? Again, assuming this is mom. What's the maternal chromosome look like? If these two were on the same chromosome. Right, dominant, dominant. Now, we talked about this last time. Mendel was really lucky in that he chose 7 characters, and they're on 7 separate chromosomes in the pea plant that only has 7 chromosomes. Right? It's a big time win. But assuming that they were on the same chromosome, the chromosome from mom would have dominant, dominant. The chromosome from dad would have recessive, recessive. And if they aren't going to sort independently, this individual can only produce gametes that have dominant, dominant, or recessive, recessive, just like we did last time. Yeah?
So when do they actually mix? When there can be like a combination?
Crossing over if they're on the same chromosome. That you'd have to have crossing over, combine the 2 chromosomes. Yeah. We'll talk about this more, don't worry. But so when you look at this cross, do you have more than just the parental phenotypes? Look at our offspring. Do we have more than just our parental phenotypes? The parental phenotypes are yellow, round, and green and wrinkled. But look at this. We have green and round, right? A little bit less creepy than the green and wrinkled. Yellow and wrinkled. A little bit less creepy than the green and wrinkled, but still really weird. I mean, it's amazing that a seed that looks like this could produce a plant, right? It just looks like something's wrong. And nothing's really wrong. It's just another phenotype. It's another flavor. All right? So if we are producing phenotypes that didn't exist in the parents, well, this would be the grandparents now, but didn't exist at any point, then these 2 genes are assorting independently. They aren't traveling as a unit. Instead, you're getting all combinations possible. So then why is our dominant, dominant still the most common form? Dominant, dominant, so yellow, round, this is dominant, dominant, right? So here are 16 genotypes, and 1, 2, 3, 4, 5, 6, 7, 8, 9 of them are dominant phenotype for both. So if they're assorting independently, why are you still having most individuals carrying dominant, dominant? Because all you need is one to make that expressed, right? And so this individual has 2 dominant, but this individual here only has 1. But it's still going to demonstrate the dominant because all you need is 1. All you need is 1. And only 1 out of every 16 is going to demonstrate the recessive for both because you have to have 2 copies of the recessive. So I told you that you don't have to do a dihybrid cross if you don't want to. Okay? Because instead, what you can do is just do 2 monohybrid crosses and then multiply them together. But it does require you to do a little bit of algebra. And if you don't like foiling things, then just go ahead and draw yourself a Punnett Square. Or memorize the ratios. And so the phenotypic ratios in this dihybrid cross are 9, which would be dominant for both, to 3, which is dominant for one but recessive to the other. You see that? So this has dominant seed shape but recessive seed color. This has dominant seed color but recessive seed shape. And this is recessive seed color and recessive seed shape. And so the phenotypic ratios in a dihybrid cross are 9 to 3 to 3 to 1. So you can memorize that, or you can draw a dihybrid cross, or you could do 2 monohybrid crosses and then foil them. Let me show you how to do that, and then we'll sum this up. All right? So what are the phenotypic ratios in a monohybrid cross? How many of every 4 offspring are going to have the dominant phenotype? 3, right? And 1 out of every 4 is going to have the recessive phenotype. So let's say we did a monohybrid cross just for seed color. Okay? A monohybrid cross for seed color. So that means both parents are heterozygous for seed color. They have 1 yellow copy, 1 green copy. But all you need is 1 yellow copy for you to produce yellow. You need 2 greens to produce green. Okay, and so the phenotypic ratios are going to be 3 yellow for 1 green. Right? And then let's say that we did a monohybrid cross for seed texture. Or seed shape, whatever you want to call it, seed texture. And again, our ratios are going to be 3 to 1, right? Mm-hmm. And normal dominant seed texture is round, so 3 round plus 1 dimpled. They call it wrinkled, but it's not really wrinkled. It's more dimpled, like a golf ball, right? Not wrinkled like a Shar-Pei. You like that reference, don't you? That was good. All right, so now if you want to do this, and you're going to combine your 2 monohybrid crosses, you multiply them together, right? And now we got ourselves a little foil, what is it, experience, maybe? So 3 yellow times 3 round, right? We do first, and then we do outside, and then we do inside, and then we do last, right? Foil. 7th grade. Or maybe 11th grade, right? Depending on how things worked out. And so 3 times 3 is 9. We should have 9 with yellow round, right? And then we're going to do outside. And so we're going to do plus outside would be 3 times 1 is 3. So 3 yellow dimpled. Wow, I don't know what happened there. And then we're going to do inside, and 3 times 1 is 3. 3 green round. And then finally we'll do last, which is 1 times 1, which is 1 green dimpled. And looky there. How many yellow round are there? 9. Look at that. What about yellow dimpled? What about green round? What about green dimpled? Look at that. Got to bring algebra back, right? You're like, I just took Gen Chem midterm. Algebra has never gone anywhere, right? Leave me alone. I don't want to deal with it anymore. All right. So what is independent assortment? The principle of independent assortment, or the law of independent assortment, says that traits of different characters come independently. They do not travel as units. They aren't inherited together. Just because the original parent was true breeding doesn't mean that those are going to be inherited together. So Mendel did this by doing 2 character crosses where he produced dihybrid individuals. So the F1 individuals, they were dihybrids. They were heterozygous at both genes, or heterozygous for both characters. And you produced offspring that had never been found before. Right? Offspring that had combinations that didn't exist before. This yellow dimpled, it didn't exist before. This green round, it didn't exist before. These 2 did, but these 2 never existed before. All right? We like it?
Yes.
I mean, you can hate it. That's fine. Just feel something. Right? Just feel something. If you feel nothing, you're not working hard enough. You can't be apathetic. Right? If you're apathetic, it means you're not working hard enough. It's okay if you hate it. I can't tell you how many classes I took that I absolutely hated. It's okay. I get it. Okay? Just feel something. Just feel something. All right? And so the recessive traits reappear, and they combine with some of the dominant traits into phenotypic ratios of 9 individuals with dominant for both, 3 individuals with dominant for the first, recessive for the second, 3 with recessive for the first, dominant for the second, and then 1 for recessive for both. Now, why can't we combine these 2 like we did in our coin flipping example, or in our law of segregation example? Right, they are not 2 different ways to produce the same thing. They are not the same thing, right? Green dimpled is not the same as yellow. Green round is not the same thing as yellow dimpled, right? They are different phenotypes. So you can't combine them like you could in the law of segregation. And that's what makes dihybrid crosses so much more fun. And then so what about, just thinking through this, if you were going to do a trihybrid cross? So you could do this with a Punnett Square. Let's go back to our Punnett Square. And you could do this with a Punnett Square. And now you're going to have, for each individual, you're going to have 16 different combinations that you could make. No, that's not true. That's not true. Take it back. I'm taking it back. For each individual, you're going to have 8 different combinations that you could make. Because you have 4, and then you're just adding another character that has 2 different options, and 4 times 2 is 8. So you have 2 parents that each have 8 different gametes that they can make. 8 times 8 is what? 64 different combinations in offspring. Who wants to do that? Right? A Punnett Square where you have 64 different combinations. But look at this. You can do it this way simpler. Because all you would do is you would go in and you would take this, right? We've already foiled this. We love it. And all you would do is go in and multiply a different character. Like let's say you wanted to do flower color, right? Flower color in a monohybrid cross, you should have 3 purple flowers for every 1 white flower, right? Plus 1 white. And then you just multiply these out. So you multiply 3 purple times each of these 4. And then you multiply 1 white by each of those 4. And you've got yourself your phenotypic ratios. And what you would find is your phenotypic ratios would be 27 to 9 to 9 to 3 to 9 to 3 to 3 to 1. 27 to 9 to 9 to 3 to 9 to 3 to 3 to 1. Or you could do, if you want to put all the 9s together, you could do 27 to 9 to 9 to 9 to 3 to 3 to 3 to 1. And then if you did a tetrahybrid cross, it'd be 64 to 27 to 27 to 27 to 9 to 9 to 9 to 9 to 3 to 3 to 3 to 3 to 1.
Is there a pattern that you're using to memorize those?
Yes. Yes, there is a pattern. Yeah, there is a pattern.
Okay. And what's the pattern?
The pattern is squares.
Okay.
So 2 squared, 3 squared, 4 squared. And then, or 3 cubed. Yeah, so there is a pattern. But it's there somewhere. All right? Well, we have 1 minute left, but, let's just ride out, into the weekend while things are going well. And things may not be going all that well.